So I'm just going to take control of the screen and find my lecture slides with any luck. It's coming up, and I'll put that onto the slideshow. And there we are. So today I'm going to be talking about characterising chromite ore processing residue urban sites. Last week we talked a little bit about differences between agricultural and urban sites, and the sites that I'm going to be talking about are shown here today. Some views from the 1900s, and on the right hand side views from just before these chemical works closed in the 1960s. This is an area of Glasgow which was very heavily industrialised. This is typically true of the Glasgow area, with a lot of the development taking place in as early as the 1770s. And a major period of their development was between the 1770s and the 1830s. This included things like shipbuilding, textiles, the iron workings and coal minings. And this map here from the British Geological Survey shows the industrial usage of many sites right through to the current time. Now, one of the things that makes it quite difficult for us when studying these sites now is that they have been used for a number of purposes. And the example that I have here is for the chemical works, which is in the area in the red rectangle. It was originally a soap manufacturer until 1810, but it became a chromium chemicals factory in the 1820s to the 1830s. So multiple uses of the same site, making it difficult for us to study these. One of the other things that is a complication nowadays is that sites may look very different. So on the left-hand side, we have the modern day picture here. This is a Google map from recent years, the scale here down at the bottom. And so you can see that there are now houses and different road structures, infrastructure here, compared to the picture from the middle of the 20th century. So this was a picture from the 1950s. So we have the River Clyde up at the top of the picture, and then we have the chemical manufacturer plant here in the orange area, and a very large waste disposal site next to the factory. And you can see that the waste hasn't even stayed within that. So there's waste that has seeped underneath the railway line here. So because the site looks very different now, it's very important for us to have historical maps and also to have information from, if possible, people who worked at the industry. This particular photograph came from one of the workers. We were given this in the 1990s. So that's very helpful when we start to investigate these sites. Now, the chromium contamination, you can see, is quite extensive, because we know that there is a lot of hexavalent chromium there. There is significant environmental concern, obviously, because chromium-6 is a known lung carcinogen, and it's also relatively mobile in the terrestrial environment. I should stress, however, that we shouldn't ignore chromium-3, which makes up a lot of the rest of that waste material. It is an essential trace element, and it's much less mobile than chromium-6, but it is still toxic at high levels. And I've just given an example here in terms of its toxicity to earthworms, with the lethal concentration for 50% of the earthworms being around 1,600 milligrams per kilogram. Now, in terms of human health in the UK, there has been a development of a model called the Contaminated Land Exposure Assessment model, and there have been soil guideline values which have been developed on the basis of this. And I've just put here for the values for total chromium for different types of land use. So you can see values here for residential with homegrown food at around about 130 milligrams per kilogram, right through to the commercial industrial sites that the safe level is 2,000 milligrams per kilogram. There are also some values for chromium-6, and these are typically much lower because of the difference in the level of toxicity between chrome-6 and chrome-3. Now, these we'll just use as indicative values. There is still some debate about the guideline values, and indeed for some elements the values have been withdrawn. But it gives us a good comparison point when we look at the extent of contamination, and I'll show you some values later on. Now, following on from the earlier contaminated land legislation in the UK, British Geological Survey were tasked with determining elemental distributions in urban soils. Previously, they had been mainly looking at rural areas and stream sediment maps and things like that. But in the 1990s and into the early 2000s, they looked at a number of cities, and Glasgow was one of those. So I'm going to be showing you some of the data from that. The survey is quite extensive. We have about 1,400 soil samples from the urban environment, and a good number also from proximal rural soils, which have the same geology, so we have the same background levels of metals, and it's a good point of comparison with the urban soil samples. For comparison with what we were talking about last day, here's the soil sampling strategy, so 5 to 20 centimetres for topsoil. They also looked a little bit deeper, only down to 50 centimetres. I think we were talking about down to 75, 76 centimetres last week. This was done by collecting augers, 5 samples per site, which were mixed together, and then the elemental concentrations determined using XRF. I'm not going to show you all the data because we have a very extensive data set that is available publicly, but the main point here that I wanted to bring out was that the Glasgow soils had much higher levels of chromium than other UK cities. In general, we tend to find that many of the heavier metals, lead, zinc and so on, were also elevated in comparison to the rural soils with a similar geology, and we especially found elevated levels in industrial and derelict sites. There were a number of other markers that we could use to pick out sites that were contaminated, and these include elements such as calcium, barium, strontium, also elevated pH. These were typical of the mid-ground areas of Glasgow, and this is the data here for calcium. A lot of this was down to the use of calcareous fill materials. And here's the full data set for calcium expressed as calcium oxide. And the area in the yellow rectangle is the area that I showed you in terms of where the chromium works are. And you can see that some of the highest calcium concentrations are in that area. All the data is shown down the right-hand side here with the percentile distributions, and the darker colours here showing that we have percent levels of calcium up to about 18%. Now, I said that could be used as a marker for industrial contamination. Well, here's an excerpt from the report for each site. So this is showing the major element here, the calcium oxide, and the other things that were also elevated at that particular site. And this one was from the former chromium chemical works. So we have about up to 20% by weight, even in the deeper sections being calcium oxide, and you can see similar elevated values for magnesium. In this study, the J&J White's chromium chemical works, that was the highest pH of all 1,400 soils, round to about 8.9 in terms of the pH. So quite elevated in comparison to Glasgow soils, which are typically pH of around 4.2 to 6. So generally acidic, where we're now going alkaline. And here we have the map for chromium, and if we were able to overlay this with the map for calcium, there's quite a strong correlation. So the calcium picks out the chromium contaminated sites quite well here. And you can see values, again, shown in the percentile ranges here, with about 10% of these sites being above the CLIA guidelines. That's total chromium, 130 milligrams per kilogram for residential with homegrown produce. So quite a significant number of these are actually residential sites. But you can also see that for the top range here, we are actually above the value for the limit for industrial and commercial sites too. So heavy contamination of quite a significant part of the centre of industrial Glasgow. And not surprisingly, when we started to do some groundwater work, we found that the boreholes were picking up quite easily the chromium-6 in the groundwaters, and this is being shown here, collection of the sample. I think we lost the first tube because these waters were extremely high pH, around about 13, and the synthetic string that we were using, I think you can see some of the string here, it seemed to disintegrate when we put the original tube into the hole. So this was our second attempt, and you can see the lovely yellow colour here for the hexavalent chromium. And this sample was from about 9 meters depth under the ground. So the sampling, so the contamination is quite deep. It's not just surface level. And these results here show a summary of all of the groundwater sampling that we did. We collected some soil solutions as well, and some surface water samples. And so you can see here that we have chromium total values for these samples up to about 125 milligrams per litre. Now that's heavy contamination, and just for comparison so that you can see the safe limit, so we have the WHO drinking water limit of 0.05 milligrams per litre. So this is a significant elevation of total chromium. And you can see also the elevated pH values for these waters, generally for the soil solution in the range 11 to 12.5, for the groundwaters, depending on where they were, from neutral up to 12.5. So it's not unsurprising that when we look at the comparison between chromium-6 and chromium total, that we find that there's general similarity. So we have very high pH, and if you look at the PE-pH diagram here, we have oxic waters, high pH, so the expected speciation would be chromate in solution. So for the surface water and groundwaters, it's almost entirely chromium-6. The exception here for the soil solution is where we have high soil organic matter content, and in these cases it dissolves up because of the high pH, but it also converts some of the chrome-6 to chrome-3, and so that accounted for the discrepancy between chrome-6 and chrome total in this case. But the big picture is heavy contamination with chromium-6 and very high pH in the waters. Here is a picture of the waste material where it has been used for terracing of a junior football ground. And what I'm showing you is what happens when, in this enlargement here, when rainwater comes into contact with the surface of that material. And so you can see that the chrome-6 has been dissolving up, and then we have the yellow precipitate on the surface here. Now these materials are up to about 4% by weight of total chromium, with about a quarter of that being chromium-6. And again, the material has a very, very high pH. And I just put down here a reminder of our indicative values for industrial and commercial land. The values will be 0.2% total chromium and only 49 milligrams per kilogram of chromium-6. Given that this is used by children for football, but they also play in this material. This represents quite a significant hazard and something that we need to think about. How long it's going to represent a hazard and what we can do about it. So that was the start of our problem. And one of the first things that we were asking ourselves was, why is this still a problem? Why are we still seeing chromium-6 in our groundwaters? And why are we seeing it on the surface of these waste materials? I've shown you here some lovely pictures of Glasgow on a good day. This is the motorway between Glasgow and Edinburgh, showing typical rainy weather, another one of the Glasgow street, and this is a very recent installation of stormwater system. So Glasgow has a relatively high rainfall with about 170 days with more than 1 millimetre of rain, much wetter on the west coast than through here in Edinburgh. So you can see there the comparison. So given that the waste material has been in these sites for 40 to 50 years, we wondered why chromium-6 is still leaching out into not only the groundwater system, but is detectable in the River Clyde. So we started by thinking about the processes that could be controlling the chromium mobility. And this slide here just summarises the main ones, and this is based on our search of the literature for chromium-6 and for chromium-3. And we considered 3 of these processes as being the most likely in terms of controlling chromium mobility at our sites. And these 3 were the desorption of chromium-6 from surfaces, the oxidation of chromium-3 to chromium-6, and the dissolution of chromium-6 containing mineral phases. So I'll just go through some of the things that we thought about. Taking the first of these, the absorption-desorption processes, we know that we have a very high pH system, and so the types of surfaces that might be responsible for holding on to chromium-6, these would be things like iron oxides and aluminium oxides. By the time we reach pH 11, these will be negatively charged, and so it's very unlikely that they would represent a surface from which then chromium-6 could be desorbed. The next process was the oxidation process. We know that we have oxidizing conditions, we know that we have very high pH, and therefore chromium-6 would be the main species that is stable under these conditions. But we also know that the oxidation of chromium-3 in the presence of oxygen is extremely slow. So that's unlikely to be happening in the natural environment under the conditions that we have. The other source of chromium-3 oxidation that's been reported in the literature as early as the 1990s by Bruce James was manganese-4 oxide-driven oxidation. But again, considering the pH of the system, we know that this process is severely restricted at high pH. And so what we're left with is that the most likely explanation for the chromium-6 continuing to leach out is via the dissolution of chromium-6 containing phases. So the next thing we needed to know was, what are these phases? We don't know an awful lot about this waste material when we started off this project. So we took a step back and we thought about the materials that were being used. First of all, the chromite ore. And this shows pictures of the ore here. And in some cases, it's present as nodules. Here, chromite ore surrounded by other material, the serpentinite material shown around about the chromite in this particular case. And on the right, we also have the structure of the chromite. So here we have an iron chromite, where we have the iron 2 present as the green tetrahedra, and the chromium-3, there's the dark blue in octahedral sites. So this material, the chromite or the spinel structure, is also found with various other substitutions as well. So it's not just a pure iron chromate that we find. There can be magnesium in these sites, and as I say, there can also be a whole range of other associated minerals that are occurring along with the ore material. But when we represent the process that's actually happening, we take the simpler structure here of the iron chromate, so that's FeCr2O4, and I'll take you through what happens in the process that converts the chromium 3 to the chromium 6. This picture here shows the site, and these are, to the best of my knowledge, the kilns where the chromite ore was placed in it. And then it was reacted with sodium carbonate in the presence of air and converted to a molten mixture of sodium chromate. So that was a roasting process at greater than 1,000 degrees. And you can see some of the byproducts being illustrated here in the balanced equation. Just as an aside, the workers who were employed by J&J Whites were often referred to as either White's canaries or White's dead men. The reason for that was they were often covered in the white material. Part of their job was to clean out these kilns ready for the next batch of chromite ore going in. And they didn't live for very long and suffered from quite a wide range of disorders. The things that we now associate with exposure to chromium 6. But going back to the process, the initial process, as I said, was with sodium carbonate, but it was found that this wasn't particularly efficient because of the viscosity of the molten sodium chromate mixture. And to counter that, there were various diluents added, lime and dolomite shown here, and these ensured sufficient air getting into the mixture and a much better conversion of chrome 3 to chrome 6. And the addition of these materials, the lime and so on, these are what gave rise to the name of the high lime process. And these also feature very highly in giving the waste materials their highly alkaline characteristics. So the next thing was done, which is important in terms of the nature of the weathering of the waste products, is that the material was washed with water to remove the soluble chromate salts. So what we're looking at is things that are not soluble under these conditions, and at the end of the day producing 2.5 million tonnes of a very highly alkaline waste residue. So that looks a bit like this. This is one of the examples of the processing residue that we collected from our sites. And you can see it's a cement-like material with white, grey and yellow colorations. The yellow is chromium 6, but some of the grey and white areas are also very chromium-rich materials. So it was this material that we then went on to characterise, and I'll take you through how we did that. The first thing we did was to powder the material and carry out X-ray diffraction analysis. And the traces here on the left are examples of COPR from 3 different sites. So it's 3 different traces that we have, but there's very common features occurring in most of them. And we go to a database then to start to work out what the various peaks might be. In some cases, we had to look a bit further afield to identify some of the minerals, but many of these we did, find in standard databases. So that gives us qualitative identification of the key minerals, but what we were looking for was quantitative information. And so a fitting process is then carried out using the Rietveld method and taking the signature for individual minerals and then summing them all together. And so we have this calculated spectrum, which is the one here, and we compare that with the observed spectrum. So that's one of the ones from the left-hand side here. And this is what we call an interactive refinement process. So what we're trying to do is to optimize the two lines here and to get a residual which is as close to a straight line as possible. And this is what we end up with: quantitative information about the composition of COPR. And we could split this into different categories of materials. So we have unreacted chromite at the top here, about 6% remaining. Then we have a category which are high temperature alteration products, typical things that you would expect from roasting of a material. And then there's another category of weathering products, and then I've picked out a special one of potential chromium-6 phases. So we have that based on how well that we have done that fitting process with the X-ray diffraction. But we'd also look to obtain additional information, additional evidence that we are on the right lines with that. And we use scanning electron microscopy for that purpose. So this is where we've taken the chromite ore and embedded it in a resin, and then taken polished thin sections of that material. And these are backscatter images, which we can then do point analysis of to get an elemental quantitative information from the energy dispersive X-ray component of these instruments. And so the first slide here of these pictures that I'm showing you demonstrates that we have the unreacted chromite, typically in the center of grains, which is what we would expect. And then around about it, we have this iron-rich material, which is brown millerite. So that is the high temperature alteration product. And again, that makes sense when we know what's happened to these grains. One thing we were a bit concerned about was that we were detecting periclase, which is magnesium oxide, because it tends to weather very quickly and form other minerals, as you see in the slides that follow. But one of the reasons that for the periclase remaining here is that it's also protected by a rim of material. And so that again explains its presence in our XRPD data. Here is the SEM for the weathering products. We have the periclase that I've just shown you, and the weathering product, the magnesium hydroxide, brucite. Over here we identified it too. And then we have two different forms of calcium carbonate. We have the blocky calcite, and we have the needle-like or chisel-like aragonite. And all of these products here that I'm showing you on this slide are from the lime and dolomite additions during the roasting process. So these all make sense from what we know happened to the ore during the extraction of chromium-6. If we need to find additional information, here's another technique that you can use. So this is FTIR, and this allowed us to confirm again that we have the brucite, the aragonite, and the calcite. So a further use for FTIR in the context of a very mineral-rich material. The trickiest part was to identify the chromium-containing phases. I've already talked about brown millerite. We knew that was there, and there is evidence for chromium substitution into this structure. So chromium-3 substitutes for iron and aluminium. That's well known from the literature. And brown millerite is also one of the phases that we know undergoes weathering, and there's evidence for the conversion of brown millerite to the first of the chromium-6 containing phases that I'm showing here, which is hydrocalamite. Now you might see that there's some unusual terms here in the middle of the slide in the third column, and these are the way that these phases are referred to in the cement literature. So the C refers to calcium oxide, the A to alumina, and the F to iron oxides. So there's a whole series of these phases. In the other terminology we have here, the small m and the small t, they refer to the number of sulfate groups, usually that are in interlayers and structures. So you have monosulfate and trisulfate forms. Now, the substitution patterns I'm showing here, these are ones that we found in the literature. For example, for the hydrocalamite, there had been a study done in chrome, hard chrome plating works, where the cement from the floor was analysed, and we knew it had been contaminated with chromium-6, and they showed that there was chromate present within that hydrocalamite structure. So we knew we were in the right lines here. But we looked again to the scanning electron microscopy for evidence in terms of what the material looks like, but also then of its chemical formulae. So that helped us to get at some of the structures that I'm showing you on these slides. So we can see here that we have the blocky hydrocalamite. It's also, you can sometimes see it as plates. It's a layered double hydroxide, so it's positively charged layers, as shown here. So that's these structures that are running here horizontally. They're sometimes called anionic clays, so they're a bit like clays, but inside out almost. And in the interlayers, what we have is water molecules, but you can also have various anions. And sulfate is a common anion that you can have, but the version that we have is a chromate substituted hydrocalamite. And that's the chromium tetrahedra, so chromate is tetrahedral ions, that's sitting here in the interlayers. So that was one of the chrome-6 phases that we had. Another one which was very easy to identify was etchingite, because it has these beautiful needle-like crystals, and it's a trisulfate species, and you can see here the sulfate sitting in between these columns. So these are calcium, aluminium, hydroxyl columns, and the sulfate sitting in between these. We found two forms. We had the sulfate form, and we also had a chromate-rich form. And by doing some stoichiometric calculations, we worked out that the maximum substitution of chromate for sulfate was about 50%. So what we were looking for was quantitative information here. How much chromium-6 do we have in this? Because then we can start to work out how much will leach over time into the environment. So it's very important that we understood these structures. And the final one, this is the one that we found hardest, was hydrogarnet, simply because there was very little information in the literature, and the only substitution pattern that had previously been reported was chromium-3 for iron-3 or aluminium-3 in the structure. When we put that in to the elemental information that we had and put it all together, we discovered that we had an excess of trivalent cations, so we couldn't get a charge balance for the structure. Only by allowing chromate to be present in the tetrahedral sites could we get a good stoichiometry for the hydrogarnet. And there's an example here of the chromium in the tetrahedral site. So we then looked at another way of confirming that we had got our quantitative information correct. Because we had that uncertainty with the hydrogarnet, we turned to X-ray fluorescence, which gives us elemental totals. And what I'm showing here in these bars, first of all, is the sum of all the chromium from our X-ray diffraction data. So adding up what we know about the material from XRD, and then comparing that with total chromium from XRF. And in the main, we get very, very good agreement. Yeah, there's a little bit discrepancy in site 4, but in the main, we get a very good match between our two ways of working out total chromium. Now, at site 4, there is the discrepancy, but we're fairly sure that it's not a chromium-6 phase. And the evidence that we have for that is using X-ray absorption spectroscopy. So we're using the near edge structure. We showed that there was 30 plus or minus 5% of total chromium as chromium-6. And when we add up the XRD quantitative analysis, we're within an error of that, 25% chrome-6. If we were to take out the hydrogarnet and say that is not chromium-6, then we have a major discrepancy. So we were quite confident going forward that we have good quantitative information about the chromium-6 containing phases within the COPR. So we're then able to move on to how does this material behave. So we carried out a whole sequence of batch experiments. And so what you're seeing here is the results for these. So each point represents a different experiment where we have adjusted the pH. We have two liquid to solid ratios, one where we have a ratio of 5 to 1, and then the other one is 10 to 1. And we added hydrochloric acid to each aliquot sample, taking the pH from about 11.7 down to about 2.5. There was a background electrolyte, and this I think is a 1 millimolar of sodium chloride for those of you who are interested in the experimental conditions. So you can see, first of all, the big picture of most of the chromium-6 being released in the range of pH 10 to pH 6. We're going to focus in on this top region here, where the results for the two liquid-solid ratios are very, very similar. And this is indicative that mineral solubility is the controlling factor here. So looking at this range between 10 and 12. And to investigate a little bit further, what do we think is happening during these processes? We know we've got all these different phases, but what's actually going on as we change the pH? So we used chemical equilibrium modelling. So this is thermodynamic modelling, where we put in various bits of information, thermodynamic stability constants, which we obtained from standard available programs like Manteca 2, Freak, but also from the cement literature. We have concentration data based on like what we have measured for the solutions and also for the solid phase. We can't just use the concentrations because of the high ionic strength. It's about 0.5 molar for the solutions that we were working with. So we have to use correction factors, the activity coefficients, which we calculated using the Davies equation here. And then we put all that information into the orchestra framework, which is essentially just a modelling framework where it's objects representing chemical speciation and transport. We can use it for a number of different things, but we're using it here specifically for looking at the mineral solubility. In terms of the chromium model, essentially what we're putting in is the composition of the COPR. That's what I've just shown you that we got. Information about the pH, the aqueous speciation of all the major elements, not just chromium, so everything that was measured in solution, and the ion activity products for the precipitation to solution reactions for the key mineral phases. So we tell it what's there, and we tell it what should go into solution if we're at equilibrium. And what we get out is quantitative mineralogical compositions at different sets of conditions, all the different pH values that we're looking at, and information about the leaching. So the chromium-6, how much is going into solution, what is the model predicting, and then we can compare it with what we observed from our batch experiments. So this is an example of the model output. The diamonds are the observed values. So say we're focusing on the very high pH end of things. So I'm only showing from pH 9 up to about 11.7 here. And again, we have the two different liquid-solid ratios being shown. So that's the two sets of diamonds that are being shown here. And then we build up the model by taking one phase at a time. So first of all, the hydrogarnet. Now we picked that one first because it was the one that was present in largest amounts, maybe up to about 20–25% of the COPR was hydrogarnet with our chromium-6 in it. So that seemed the obvious one to start the modelling with. However, you can see that it underestimates the leaching of chromium-6 at all the pH values. If we take something like hydrocalamite with a layered structure with the chromate in between the hydroxyl layers, then you can see that we have quite a good agreement in the range of pH from 11.7 down to about 10.3 between the model output and the actual behaviour. Now we have to take account of the other elements as well, so you can see that there are more complicated models here with some of the other minerals, but in terms of giving us the best fit, the hydrocalamite on its own seemed to give us quite good agreement. But we're not doing very well once we go below about pH 10.3. So there were various other steps taken, and one of the things that we did was to look at the relationship between hydrocalamite and one of the other key chromium-6 containing minerals, the ettringite. That was the one with the beautiful needles. From the literature, what we found was that as hydrocalamite dissolves, then typically what happens is that chromium-rich ettringite tends to precipitate. So once we allowed precipitation to be part of the model, then we see a much better fit of the data down to now about pH 9.5-ish, thereabouts, before we have the discrepancy. So in that top range of the pH, we have the ability to use thermodynamic equilibrium modelling to give us a reasonably good prediction of what's going on in the solution. Now, we've made quite a lot of assumptions because on the model we're using individual phases rather than the real mixture, which might have surface coatings on it and other factors that we'll come to in a minute. So we wanted to check that the model output agreed with what we were actually finding happening. So we looked at the residue from the batch equilibrium experiments where we're leaching chromium out, and what we're looking at here is what was left in the residue, and I'm only going to show it for the hydrogarnet and the hydrocalamite. Now, we predicted that these would dissolve up almost entirely, so that below pH 10 we were not expecting to find these phases. And what we actually find is that yes, they are dissolving, but they do tend to hang around a little bit longer and to lower pH values than we would expect. And we did these experiments over 4 days shaking and 26 days shaking. So that's why you're seeing two sets of diamonds here. For the hydrocalamite diagram, you'll see an extra set of points. And this is to do with the X-ray diffraction, where we observe the splitting of the hydrocalamite signal. We are fairly confident that it was still hydrocalamite with a different substitution pattern. So we included the extra data set here. So what we're seeing here is that there's broad agreement between the model output and what we're observing happening in our batch experiments. But there may also be some kinetic factors at play here, which are, if you like, protecting some of minerals like hydrogarnet and hydrocalamite. We went back to the scanning electron microscopy to explore why this might be happening, at least in part why this might be happening. And so I'll show you some more of the thin sections here. Here was the chromite in the middle, which is the unreacted material. And on the outside we have the brown millerite. That was the hard rim. But if you look where we find the hydrogarnet, it's inside that rim. So it's at least being in part protected by the outer coatings on these minerals. And that might be part of the explanation for the slow dissolution. On the right you have the hydrocalamite, and again there's some kind of protective material around the outside, at least of some of the hydrocalamite. So is it important to look at the pH range where we are not getting the good agreement between the modelling and the observed behaviour? Well, from our knowledge of how this material behaves, it may not be, at least on the time scale of decades. Because the other part of what we're doing is looking at the L changes in the COPR material and its resistance to change in pH. So we took some of the material and placed it in a column and passed something like 194 volumes through this material, which was equivalent to about 4 years of Glasgow rainfall. And in doing that, we were able to change the pH from about 12-ish only down to about 11.2. So the range where the model was working really well, we can understand the behaviour mainly in terms of mineral solubility. Then we're very well covered with that. So in terms of understanding the behaviour at the site without any treatment of it, then we are likely to be able to make predictions on the timescale of decades. If, however, we want to do some remediation, then we might need to look a little bit more at our understanding of what's going on at different pH ranges, but that's something that I will tackle in my later lecture in a few weeks' time. So just to sum up, I think it's fair to say that understanding contaminant behaviour at industrial sites in urban environments can represent quite a difficult challenge. If you know about the site history and if you can get information about industrial processes, that can be very useful as a starting point. When it comes to characterising, we've found that the X-ray diffraction information, particularly when we use it in quantitative mode, was very useful, but it's also quite important to get additional evidence for that interpretation. And the main techniques that we can use for that would be the scanning electron microscopy, in some cases FTIR spectroscopy, things that you have readily available in most laboratories, and then at the higher end of things, sometimes only in a few laboratories we might have access to X-ray fluorescence and X-ray absorption spectroscopy. We were able to carry out modelling. We identified that the dissolution processes were the important ones. You also have to be able to get the information in addition to your quantitative information. You need thermodynamic data. That's not always available from standard databases, and it may require scouring of the literature and some experimental work to get the necessary thermodynamic data. We were able to successfully carry out our modelling over a limited pH range using thermodynamic equilibria, but it was quite clear that there can be thermodynamic factors, and you can't overlook these when you're studying these complex systems. For checking the modelling, we were able also to use the X-ray diffraction, and the main way we did that was to use the scanning electron microscopy again. So that gave us a good understanding of the site. I don't think that what we've done is restricted just to chromium contaminated sites. It would be applicable to other scenarios where we have high temperature waste products, incinerator sites, maybe also fly ash materials, and also the nuclear industry. I've seen this approach being used. Chromium-6 contamination is an issue there in some of the waste materials, and in deep disposal sites where they're considering the use of cement backfill, the type of mineralogical information and the modelling approach here may also be very relevant. So thank you very much. I'm going to stop there, and I will also stop my share, and I'll take any questions.
Margaret, I was wondering, these rinds that form around the minerals, first of all, how pure are they, and how much do we know about their solubility, and how long, like, you say these sort of protective coatings, are they relatively a pure substance, or are they a mixture of things?
I think they're a mixture of things. I mean, the spot analysis found some very magnesium-rich components, but it also found some very iron-rich. So there's a variety of substitutions that we find for these. In terms of the longevity of the coatings, these are minerals that have been in the environment for 40, 50 years. So these rims are lasting on that time period. Any more questions?
Professor Margaret, yeah. Thank you very, very much for the talk, and I have learned a lot with all the methodology that you have used. I have a question about the organic matter that could be released at this really high pH value. Do you have any idea about the mobilization of the organic matter in your site, please?
The organic matter,
yeah.
Yes. One of the sites that we looked at, in fact, it was just behind the Rutherglen-Glencairn football ground. There was a soil that had 20–30% organic matter, and when the waters were flowing through that, the colour of the soil solution was very, very dark brown. It was almost like you had done a sodium hydroxide extraction on that material. And the chromium levels were very high, say of the order of 120 milligrams per litre, and it was all chromium-3.
Okay.
And so you have the chromium being transported away from that site. So if you have high levels of chromium-3, they can still represent a hazard. We didn't find it all sites. There were some sites where there was a mixture of chrome-6 and chrome-3, where you have small amounts of DOC in the water, maybe a few milligrams per litre. So the dark brown solution was the extreme case. More typically, we observed either the pure yellow colour or somewhere there was a small amount of DOC present.
Thank you.
Has anyone else used the orchestra model?
I have just heard about it, but yeah, I never have changed to use it. I think you have a chat question.
Oh yes, one moment.
Can you hear me?
What kind of environmental changes would create the most risk for the chromium-6 release from the minerals? Yes, a good question. I think that it's quite a hard material. So it's a very dry material, so the rainfall doesn't seem to affect it. So because it seems to be a kind of crust on the surface of the material. So I talked about crust on the surface of the individual grains, but when you go to the site, the very surface-like layers, so maybe 1–2 centimeters, that also has a different mineralogical composition, and it's actually very hard for water to penetrate that material. So the water tends to kind of go around it, and again, that's another reason why we tend to get the slow leaching. But so major changes in rainfall, I'm not sure are going to have an immediate impact on that, because the water doesn't tend to get in there. That also holds true for sort of bacterial things. So like if you're looking at remediation, it's very hard to get microbial entities in contact with it. Trying to think, you would have to have a really major source of mineral acid to have a large impact. I think if you look at the leaching data, if you were able to bring the pH down to the range between 6 and 10, then yes, that would have a massive impact. That's where a lot of the chromium would start to be leached out. But that's not really what we would find in the environment unless we've had a major spillage. So, fortunately. Okay, I'm not seeing any more questions.
May I have a question? Do you hear me?
Yes.
Hi, Margaret. Thank you very much. It was an excellent talk. A follow-up to the last question. You said in the 4-year rainfall, you had a pH decrease from something 12 to 11 point something, right?
Yes.
But the pH decrease may depend, does depend on the buffering capacity of the soil. So it may be different at lower pH. So did you do any determination of the acid
buffering capacity of the soil and how it would, the pH would develop in the future? Did you get the same input of acidity?
I'm not hearing most of that. I picked up the original part. Sorry, I'm just being given a set of headphones here. So the question was about the change in pH. So
as the buffering capacity of the soil at lower pH, would it be as high as it was in this range of 11 to 12, or would it decrease? Do you know it?
Yeah, I think the buffering capacity is much lower. Once you get past, once you're going down from the 12 through the 10, there is information about the extent of buffering in the 2002 paper, the Geochimica Cosmochimica Acta paper. And there are 2 points, I think, where you can see a major change. I think one of them's around about 10 and one's around about 8. And then after that, I don't think that the material has extensive buffering. Things do happen below that. There are major changes in speciation. We have precipitation of iron phases. And we think that the chromium-6 then gets removed onto the surface of these oxide minerals. Below pH 4, however, we have definite evidence of conversion to chromium-3, and chromium-3 then starts to shoot into solution below pH 4. So you get a large increase of chromium-3, and it may be some of the chrome-3 phases are starting to dissolve up at that point, but we didn't do too much work below neutral because of the fact that we're not observing the change in the environment, even though Glasgow soils are typically in the range of 4 to 6. These materials seem to resist that change. So if it's Glasgow rainwater that we simulated, the lowest pH that we managed to achieve was about 11.
Thank you.
Okay, so I think that's probably most of the questions for now. If there's any more questions, then you can perhaps discuss them with Hui in the short discussion afterwards, or you can email me or save them up for next week. Next week we have Rainer who's going to be speaking about the shooting range soils, and I'll be here to introduce him at the beginning of the session next week. So thank you very much for logging in today, and I look forward to seeing you again next time.
And since there's no one to say thank you to you, Margaret, today, let me thank you for a wonderful lecture.
Thank you very much, and enjoy
Thank you very much.
Bahamas.
Have a good day
Okay.
or night, everyone.
Thank you. Right, bye-bye.
Thank you. Have a nice day.